I didn't know.
Okay.
All right, continue.
So let me, so you guys still see my screen?
Yes.
Guys?
Yes, sir.
Okay. So last time we learned about the applications of biology. So today we'll be looking more in depth into the organisms. And the first one we have is bacteria. So we say that bacteria is a single-celled organism. Anyone know what single-celled mean?
Yes, it can't can make more cells.
Yes, it is only made up of one cell. So technically, a bacteria is just a cell. It's not like us humans where we're made up of like millions of cells. And these millions of cells make up our organs, and our organs make up our bodily systems. Everyone following?
Yes, sir.
And we say that we can find bacteria everywhere on Earth, so we can find them in our body. Probably on the chair that you're sitting on, you have some bacteria on there. There's some bacteria on the computer screen that you're using. There's some bacteria in your kitchen, some bacteria in your bathroom. Everywhere there are bacteria. So anyone want to tell me any place that they think bacteria can exist again? And we also say that bacteria can live in extreme temperatures and pressures. So bacteria can live in the earth core that would have extreme pressures, extreme temperatures like in the desert, that would be extremely hot. And at the North Pole, that would be extremely cold. So this is why bacteria has been around for millions of years. Everyone following so far?
Yes,
Yes,
sir.
sir.
And here we say that the human body is full of bacteria, and it's said to actually have more bacteria than actual human cells. So right now inside of your bodies, there is more bacteria than actual human cells. So there's more bacteria than the actual cells that you would need as a human being. And we say that most bacteria are harmless and some are even helpful. And we learned about that in Monday's class. So bacteria can come in 3 types of shapes. We have spheres. Anyone know what a sphere looks like? We say that the earth is a sphere. Anyone learn about the earth in class and they say that the earth is a sphere?
Yes, sir.
And we say we can have rods or spirals.
Yes, sir.
So any bacteria that is harmful to the body is called a pathogen. It's called a pathogen, and we describe it as being pathogenic. So pathogenic would be the adjective for bacteria that are harmful, and these can cause diseases. So let's look at some of the shapes. So right here we have the spheres. So we would say that they kind of look like circles, but technically not really circles. And here we have rods. So they're kind of long. And for the spirals, we have it has some curves. Everyone can see that?
It is in a column, so we'll do the job work for garments.
We can see that the spirals here are more curved than how the rods are kind of straight, while the spirals have some curves in it, and the spheres are round. So let's look at the bacterial cell.
All right, I sent a job work for.
The first part of a bacterial cell is the capsule.
Just
Everyone following? You see this capsule?
Yes, sir.
We have the plasma membrane. We can also call the plasma membrane the cell membrane. So right here, we have so first we have the capsule. then the plasma membrane, and inside, and the plasma membrane is surrounding the cytoplasm. The cytoplasm. And we also have a cell wall right here. So outside, so the cell wall is around the plasma membrane. Can we see that? We see the cell wall?
Yes, sir.
And we have ribosomes. Ribosomes are what makes proteins. Anyone know what a protein is? I'm sure you guys know what protein is.
Yes, sir.
And inside the bacteria cells. So unlike human cells have nucleus, and we say the nucleus is the what of the cell. It's like the brain of the cell. It controls how the brain interact, how the cell interacts with its surroundings. So it controls what the cell does. However, bacterial cells do not have a nucleus. They only have a single strand of DNA. So they only have the they only have the DNA, which is their genetic material. However, in a human cell, the DNA will be found in the nucleus. Hopefully, I'm not going too fast. Everyone understanding?
It's in your mouth. Go sit back here.
Everyone following so far?
Yes, sir. Yes, sir.
Yes, sir.
So where in the human cell would we find the DNA?
Stop.
Where in the human cell would we find the DNA? Anyone wants to give that a try?
What should we try?
I'm asking where in the human cell would we find the DNA? If
In
you
the
guys are-
nucleus.
Oh, okay, very good. So let's look at the functions of each part. So we say that the capsule acts as a protective coating. When you hear the word protective, what comes to mind? When you hear the word protective, what comes to mind? No one knows what it means to protect? To guard something from harm or from danger. So the capsule acts as a layer so that the cell doesn't easily die, that different chemicals won't harm the cell as much. And the capsule is what would help the bacteria cells to survive under extreme temperatures and pressures. And next we have the cell wall. So we say that the cell wall gives the cell its shape, and it also protects the cell from the outside environment. And we say that the cell has 3 different shapes. Anyone can name one cell shape? You don't have to talk. You can put it in the group. You can put it in the chat. No one wants to put it in the chat.
What is it, sir?
I'm asking you guys to name one cell shape in the chat.
Okay, sir. Should I just send it to you privately?
If you want to say it out loud or you want to put it in the chat, anyone. Okay, very good. we don't have any rectangular. We don't have it. Yes, we can say circular too. Yes, very good.
Well, a plant cell is rectangular.
Unfortunately,
A plant cell
we
is
have
rectangular.
we have rods and we have spears.
Oh, sir, a plant cell is rectangular.
What?
A plant cell is rectangular.
I didn't say plant cell. I said about a bacterial cell.
Bacteria cell.
Yeah, I'm sorry about that. Not a plant cell.
Oh, bacteria cell. Okay.
Yeah. Don't want to mess that up because that's coming on the exam. So we say that the cytoplasm, it functions for cell growth, metabolism. So the functions for cell growth and metabolism happen here. So inside the cytoplasm is where the ribosomes will create protein. So when you guys go on to high school, anyone going to high school soon? Because I think you guys are like 12 to 13.
I'm in second form now.
Okay, that's very good. So in sixth form, you guys will learn about, or in fifth form too, I see they're doing it now. You guys will learn about transcription and how the cells move DNA from the nucleus to the cytoplasm via RNA. But when that times come, you guys will learn about that. So we said that the flagellum or the flagella plural. So they are structures that resemble hair and are used as a means of locomotion or movement. Anyone learn about locomotion in their science class in high school or in grade 6 science class? We learn about senses and we learn about movement and locomotion.
The name is familiar, but I don't remember.
So we know that in humans, our legs are what we use, our feet are what cause us to move. So our legs are the instruments of locomotion in humans. But in the bacterial cell, the flagella is what they use for locomotion. Everyone understanding?
Yes,
Yes,
sir.
sir. What do you
Yes.
say, sir?
I'm saying that in humans we use our legs, or we can say our feet, as a means of locomotion or movement. So if you want to move from one place to the next, wouldn't you walk and you use your feet to walk?
Oh, yes, sir.
And so bacteria use the flagellum to move from one place to the next. And cars have their wheels. So locomotion is just movement, moving from one place to the next. Everyone following so far? Now we have the villi. As I said, that bacteria can be found everywhere on Earth. So they must need something to help them stick to those places. So we say that villi are structures that resemble tiny hairs and help bacteria to attach to other cells and surfaces. Example, the teeth, intestines, and rocks. So as we know, bacteria is in the human body. So bacteria can attach to actual human cells or any other animal cells. Bacteria can also attach to plant cells, and the villi is what they use to attach to those cells. And right now, even as I'm talking, there's bacteria on my teeth, there are bacteria in our intestines, and if you go outside and you put your hand on the rock, some bacteria uses the villi to attach to your hand. So that is why our parents tell us to wash our hands before coming inside from playing, because bacteria and some bacteria can be harmful. And for the ribosomes, you just have to know that ribosomes help in making proteins. Any structure you guys want me to go over that you weren't clear on? Anything you guys not clear on so far and you want me to go over? Don't be shy. If there's anything you guys didn't understand and you want me to repeat in simpler terms, just tell me. Everyone's good? Okay. So let's go on to viruses. So now we learned about bacteria, but now the virus is actually a harmful microorganism. So we say a virus is a small parasite that cannot reproduce by itself. When you hear the word parasite, what comes to mind? personally, for me, when I hear the word parasite, I think about unwanted. Any other words come to you guys?
Worms. What comes to mind is worms. Parasites.
What? What comes to your mind?
Something about worms.
Something about worms. Okay, worms can be parasites in some aspects, so very good. So if you don't want, if you have a plant and you don't want worms on the plant, we can say the worms are parasites because they are unwanted. Yes, very good. So let's look at the properties of viruses. So viruses have no membrane, cytoplasm, ribosomes, or other cell components. So they cannot move or grow. They can only reproduce inside a whole cell. So let me give you a little, let's talk about the COVID-19 virus, for example, or we can talk about so what the virus does is it travels inside of the body, then it sticks onto the whole cell and like use the whole cell to make copies of itself.
Like
You guys
malaria.
understand? What?
Like malaria.
Like malaria?
Yes. When malaria is injected into the bloodstream, it makes its way to the liver and tries to go to a liver cell and might destroy one or two cells on the way. Then it grows and then makes more of itself, which is then injected into the bloodstream. Then those malarias are now programmed to target blood cells, and then they go into the blood cells so that the immune system cannot touch them. And after a while, they just reproduce in the blood cells, and the blood cells get heavy and sticky, so making them stick to the blood vessel. And then the blood cells burst, and then there's more malaria. There's lots more viruses.
Okay, very good, very good. You want to become a scientist in the future? Very good.
No, I
So
want to
two
become a
major-
robotic engineer.
Oh, very good. So two major parts of the virus is the protein code, the DNA and the RNA, because it has to use DNA or RNA to actually hijack the whole cells. So we say that viruses are extremely tiny. They're smaller than than cells, and they're only visible with electron microscopes. So here they say that even light microscopes have trouble help seeing. Even light microscopes have some problems seeing viruses. So we say that the shapes of virus are predominantly of two kinds, rods or filaments. So let's look at how viruses infect people. So we say that our bodies are made up of millions of tiny cells. Viruses cannot survive outside of the body for long, so they try to get into the cells in our bodies. When this happens, we say we get infected. So the virus multiplies and can spread around the body. So that is why we cover our face with a mask, because the coronavirus goes it can go up our nose, and then there are cells in our nose, and then the virus replicates and goes into the rest of our bodies. Everyone following so far? You guys understand what I just explained?
Yes, sir.
Okay, very good. And we say our bodies then fight the infection by producing antibodies which help us to get better. However, not every virus will be affected by antibiotics. We said that some viruses have antibiotic resistance. That means that they can put up a good fight against the antibodies that your body produces. So if anyone has ever been with sick yet, so the first time as a baby when you have the flu, your body is going to make antibodies to fight it off. And the next time you're going to get the flu again, your body has already know what antibodies to produce, so you can fight off the flu faster. So the first time we catch the flu, a virus enters our body, the virus replicates and the body produces antibodies to help us fight that virus. And the next time we catch the same flu, our body already has antibodies, so it helps. So that's why we get better. That's why we get better faster the second time that we catch the same cold or flu. Everyone understanding? No, yes, maybe so.
Yes, sir.
So we say when viruses enter our body, they can stop normal cells from carrying out their important bodily functions. So what we can say is that the COVID-19 virus what do you think the COVID-19 virus is stopping? What kind of bodily function is the COVID-19 virus stopping? So when people say they get the COVID, they find it hard to breathe. So the lungs are having troubles breathing. So the lungs have trouble exhaling and inhaling oxygen. So it's stopping respiratory functions in our body. And we say we can protect ourselves against viruses by washing our hands with soap and water. Don't forget the soap. So if you wash your hand with water alone, the virus cannot be killed. And if you wash your hand with soap alone, you have nothing to wash away the viruses when they die. Everyone following? So let's look at how the soap works. I don't have it. I don't have how soap kills. All right, here is it. So the coronavirus has a membrane of oily lipid molecules. So we say the oily lipid molecule, what we call the protein coating. See there, it's the protein coating, and it is what helps the virus infect cells. However, soap molecules have a hybrid structure. Hybrid means two different parts. So it has a part that bonds to water, and it has a part that hates water. And here we say how the soap destroys the virus. Sorry about that. So when the water shunning or the water hating tails, or the water hate, we say we can't call the tail. So we say that the tail is hydrophobic, and hydrophobic means it hates water. It doesn't want to come near to water at all. And hydrophilic means that it loves water. It always wants to be in the presence of water. So what the tails of the soap molecules do is wedge themselves into the lipid membrane and pry it apart. And we say that the soap traps dirt and fragments of destroyed virus in tiny bubbles called microcells, which wash away in water. So that is why we need water. And if you don't have water to wash away those tiny bubbles, you still actually have components of the viruses on your hand, of the virus on your hand. So what the soap do is pull the COVID virus apart. You guys following so far? So the hydrophobic tails latches onto the outer layer of the cell, and that is the protein layer, the layer that is made up of oil, and it pulls it apart. When you add the water, the soap starts to pull it apart because the hydrophobic tail is trying to get away from the water. Has anyone tried the experiment where you put oil and water in a cup? Anyone ever tried that before?
Yes, sir.
And what happens to the oil?
It floated.
It floats on the top of the water because oil is hydrophobic. Oil hates Oil hates water, oil and water does not mix. And that is what is happening to the outer layer of the virus, of the coronavirus. So that outer layer that is made up of oil attaches with the outer layer, attaches with the hydrophobic part of the soap, and they try to break away from water. So they don't want to mix with the water. And that is what causes the cell to burst into the tiny bubbles. Everyone understanding? That is what causes cells to move away from the water. So let's look at how viruses enter the body. So we say that the virus enter the body releasing RNA. Let me make it bigger for you guys. Virus RNA invades the cell nucleus and takes it over. So it's technically like a hacker. Anyone ever had a virus on your computer before, and you see how the virus causes your computer to do things that you don't want the computer to do?
Sure.
So in this case, let's say that you are the brain of the computer. You're the person that is controlling the computer. However, unfortunately, you do something and a virus enters the computer. What the virus completely ignores your instructions. The computer completely starts ignoring your instructions and start doing the instructions of the virus. And this is what we say is happening inside of the body. The virus enters, invades, takes over, and your body cells stop doing what the nucleus of them, what their brains tell them. Stop doing what their brain is asking them to do. So we can say the body cells start getting unruly. So we say viral RNA uses the host cell to create new RNA and assemble more viral particles. So what is happening here? The viral RNA tells the host cell that, look, you have to start creating more of me, or in standard English, it tells the host cells to start creating new RNA and start making more virus. And then new virus particles are released, sometimes destroying the cell in process, unfortunately. And this is why viruses are very dangerous. And we say that RNA is known as ribonucleic acid. Anyone can pronounce that word? Ribonucleic acid? Okay. Only have a few minutes left in class, sorry. So let's look at plankton. Anyone know plankton from SpongeBob? Anyone watch SpongeBob? I know plankton.
Yes.
Yes,
Yes.
sir.
Yeah.
Let's look.
Yes, sir.
Okay, very good. So we say planktons are very important in many ecosystems, from providing food for biwaves to live. So there are two main types of plankton. We have phytoplankton, which are plants, and zooplankton, which are animals. And we can remember that a zoo keeps animals, so that is how we can remember what a zooplankton is. So a phytoplankton plant and a zooplankton animal. And we say that we keep animals in zoo, so we can remember it that way. So we say zooplankton and other small marine creatures eat phytoplankton and then become food for fish, crustaceans, and other large species. So we know that plants make energy during photosynthesis. Anyone know about the process of photosynthesis?
Yes, I know.
Or how plants use the green
Yes, sir.
Okay, so plants use the green pigment inside of their leaves to capture sunlight and use it to make energy, and then they release oxygen, and we as humans use that oxygen to breathe. So actually, phytoplankton make their energy through photosynthesis, the process of using chlorophyll, which is the green pigment in plants, and sunlight to create energy. So like other plants, Phytoplankton take in carbon dioxide and release oxygen. And we say that phytoplankton account for about half of the photosynthesis on the planet, making them one of the world's most important producers of oxygen. So planktons are actually very important. And we say in addition to phytoplankton and zooplankton, two even smaller kinds of plankton can be found floating in the sea that are bacterial plankton, which are bacteria, and viroplankton, which are viruses. And so planktons can be found in salt water and fresh water. And one way to tell if a body of water has a large number of plankton is to look at this clarity. Have anyone went to any place that they see how the water is dirty and murky? You ever go to any lakes, rivers, and the water looks greenish?
What you say? What you asking, sir? I can't hear you. Breaking up.
The water is not clean.
What you say,
Yes?
sir? What did you say, sir?
I'm saying that if any of you guys have ever went to any lakes or
What
any
you say,
rivers,
sir?
you're not hearing me?
No, sir. Can you say it again, please?
I'm saying that has anyone in here ever went to any lakes, rivers, ponds and see how the water is dirty?
Yeah, I've been to a river and I saw that the water was kind of green in one part.
Yes, in part. So so they're saying here that possibly that water has plankton because water change because they're in such a huge variety that they start changing how clear the water looks.
Yeah, but it didn't stay for long, sir. It just went down the current.
So here it says very clear water usually has less plankton than water that is more green or brown in color. And here we have how some of these look, so you can go over that and look at them.
So, sir, you saying if we like swallowed river water by accident and it was green, we could have eaten plankton or something?
Yes, that is what I'm saying. You're more likely to have ingested some plankton.
Okay.
And let's look at fungi, or fungi, plural. So fungi are more closely related to animals than plants. Fungi are heterotrophic. Heterotrophic, they're like human beings. That means they don't produce their own food. They depend on plants to produce food and we eat them. So as a human being, we depend on plants to produce food, and the food has energy, and then we eat the plant and we get energy from that plant. Or we wait on the next animal to eat the plant, and we take the energy from that animal when we eat them. Everyone following so far?
Yes, sir.
Yes, sir.
I will say fungi multiply after asexually, sexually are both. I'm not sure if should I be explaining this concept to you guys at this point? So asexual means that they don't need any parent. They only need one parent. So that one parent can produce by themselves. And sexually means that they need two parents. So they need a mother and a father to help to produce new fungi. Everyone understanding?
Yes, sir.
So asexual, you only need the mother or you only need the father, and you can produce more fungi. Sexual, you need both mother and father. So humans reproduce sexually. That means you need your mother and you need your father to make new human. So majority of fungi produce spores, which are defined as multicellular. So we say that fungi are multicellular. And we say that fungi interact with other organisms by either forming beneficial or mutualistic associations or by causing serious infections. So have you guys learned about mutualism? I think that is one that you should learn about that in one of the classes.
Yes, sir, we did.
Okay, very. And what do we say mutualism is? Anyone want to say what mutualism is?
Isn't it when two organisms basically benefit each other? Provide some form of benefit.
Yeah, very good, very good. Very good. So let's look at some typical fungal cells. So they have a cell wall, they have a cell membrane, they have mitochondria, they have cytoplasm, they have a vacuole, and they have a nucleus. How do they differ from bacterial cells? Anyone can tell me how they differ from a bacterial cell? What does this cell have that the
because they
okay,
because
very
this
good. And it doesn't
because
have the mitochondria.
yes, doesn't have a mind. No, it's not that. It's that bacterial cells. Is that bacterial cells more have a tail, like have tails.
Yes, but and they also don't have a nucleus as we discussed earlier. So you're right on both parts. Is that okay? Is that okay with you? You're right on both parts? So let's look at host-parasite relationships. We only have 1 minute. So a parasitic relationship is one in which one organism, the parasite, lives off another organism, the host, harming it and probably causing death. So the parasite lives on or in the body of the host. Anyone know any parasites in any common parasites? Anyone know any parasites that lives in
The parasite.
like worms? You know that they say we have worms inside of us. We have intestinal worms that live inside the intestine. And in Jamaica, your parents go outside and buy Zentel to kill them. So those intestinal worms are actually parasites, and they can eventually harm us if we don't get rid of them as early as possible. So let's look at factors that determine the outcome of most host-parasite relationships. So we say that specific factors influencing host-parasite associations are among climate conditions, behavioral traits, host sociality, population density, diet, habitat, age, sex, host immune health, supplementary feeding, and animal translocation or movements. Let's say we have let's say we have cattle, or we have cows. The cows are moving from one place to another. It's more likely that the cows can pick up some type of parasite and have them in, and then they
Animal parasite.
they ingest these parasites, and then they make the cow meat harmful for us as humans to eat. Anyone eat cow? Anyone eat beef? Anyone in here eat beef?
Sometimes.
Yes, sir.
The evolution of host-parasite relationships. So we say that most parasitic relationships are very specific and complex. And what that means that each relationship, that means that the relationship between a dog and his parasite won't be the same as the relationship between a cat and her or his parasite. So each is specific and each is very complex. So there's a range of different interactions. How a parasite affects a human being won't be the same way a parasite affects an animal. How a parasite affects me won't be the same way it affects you, since I have a more mature immune system than you guys at this point in life. So I will be better capable of warding off parasites than you. So that's why we say that the relationships are very specific and complex. You guys understand that? And then we say the parasite and host have co-evolved. So what we're saying that along with humans, parasites has evolved with us. So if you evolve, the parasite can also evolve. This means that the host has developed a defense mechanism. So the immune system are hydrochloric acid in the stomach. And you know what anyone know what hydrochloric acid is? We call it HCl in chemistry class.
No, sir.
Okay. You can look at that up more. So it's a chemical.
It's
So
like it's like the acid that breaks down food.
Yes, very good. Yes, the acid that breaks down food. So the host, so humans have, so what that acid does is like kill certain parasites that is on the food. So that is what makes most, because not all food, even if you wash some types of food, that doesn't mean that the parasite on that food is dead. So the body has hydrochloric acid to help fight off any parasite that may enter the body. And we say that the longer the relationship has existed, the more host specific the parasite becomes. The more host specific the parasite becomes. So the longer you have intestinal worms, the more specific those intestinal worms becomes to your body, the more harmful it becomes to you. You guys follow me? That is all we have for today's class. You guys can start leaving now.
Thank you for teaching us, sir.
Yes? Yes, who was talking? Class time is finished. Anyone want to ask any question before they go? I went about 5 minutes over time. I'm sorry about that.
Yes, sir. Where are we to go next?
I'm unsure.
Oh,
Let me look at the schedule and advise you guys.
Guys, good morning. Good morning again, everybody. I will assign you to your different rooms. I'm just waiting on your tutors to arrive on the platform.
So you guys have long division now.
what about for tropical forest, sir?
You have aseptic techniques. Just give me a second, please.
Yes, long division. I'm ready to go whenever we're ready.
Okay.
Yeah.
measurements that we did the first day. But before then, because I remember that I think there was a few of us who didn't really understand. Oh yes, the recording session, yes. So there was a few of us who did not fully understand the session before. Give me one moment, let me see here. All right, perfect. So yeah, because there was a few of us who didn't fully understand the session before, I want to go ahead and start with another little video that should summarize everything that we did yesterday, and then just spend the rest of the time working problems and then going over the answers. How does that sound? Sounds great?
I'm not.
Pardon me?
Yes, sir.
All right, perfect, perfect, perfect. Yes, thank you. Yes, recording the session now. All right, here we go. So we're gonna start with another, we're not gonna start with a song this time, but I hope we can remember from last time that the 4 basic steps of long division would be to
Tyrese, you should be in. Please join the Tropical Forest group. I assigned you guys to the breakout room already. Cumberland, are you having any problems joining the wetlands group? Good morning, everybody. You're supposed to be having aseptic techniques in lab right now. Your tutor is having some technical difficulties coming in terms of getting on the platform. So just give us maybe 2 minutes. Sorry about that.
Okay, sir.
All right, guys. So your tutor will be on shortly. With the internet, we have technical difficulties. Continuing the discussion until he gets here. That we were having this morning. I was saying that you guys need, especially with the global environmental issues sessions coming up next week, you guys need to be up for the activities planned. You need to be abreast, keep abreast in terms of environmental news. Ram Prasad, are you aware of the new legislature regarding waste disposal in Trinidad that was signed this week? Hello? I guess she's off. Rajwani, did you see the clip about the Rio Grande, the Rio Cobra on TVJ on Tuesday night?
About how there's an overkilling of fish in the Rio Cobra?
Yes.
Yes, that's it, sir.
Okay, what did you think? What do you think are the impacts on the ecosystem?
There would be a large impact on the ecosystem and even the people who are living there. One, the ecosystem is because the fish living in that river are necessary for animals, as there were food source for animals, and also the welfare of the Rio Cobra, as many fish eat the dirt and eat the many harmful bacteria in the water. But if there's no fish in the water will remain murky, dirty, and unhealthy, and that will, force humans to move from the recovery, and the recovery will eventually become unusable.
Okay, good. We'll discuss it more in the third week when you have water chemistry together. All right, Mr. Morris is on, so you guys have his session now.
Hello, good morning, everyone.
Good morning.
How is everyone doing this morning?
Good.
Your morning has been good so far.
Yes, sir. How about you?
It's been a rough start, but we'll see how we can get over that this morning and try to pull off our session. So today we're going to be talking about aseptic techniques in labs. Now, when you hear the word aseptic, what comes to mind? Anyone?
I do not.
Repeat that.
I do not know.
Okay, that's okay. So aseptic techniques would refer to any measures in labs that you would take to prevent, yes, for example, contamination and other things that will mess with the possible results of your experiment. So any measures that would you would take to prevent your techniques from being contaminated and from developing a yeast or a mold. Has anyone ever seen a yeast before or a mold? Like you might have seen it on cheese, that greeny good, yeah. So there's a slight difference between a yeast and a mold, and I want us to watch a video on that. So I'm going to try and share screen now. Okay. Is everyone hearing?
Sir, we can't hear the audio.
Oh, you can't hear? Okay, give me a second. Okay. Give me a second. Okay, and so I'm just waiting on your coordinator to try and share it.
Mr. Morris?
Yes.
Yeah, you hear me, right? Yeah. Yes. Just when you click share screen, there's a small tab that comes up. After you click the specific window that you want to share that says share sound and optimize for video clip, you can just click those two tabs and it should work fine.
Okay. I'm not seeing it. I'm not seeing it, so I'm gonna try something else because I'm not seeing that option.
You want to send me the link for the video and I play it?
Okay. So I'm going to send you the link now. Okay, so while Sarah is trying to play the video. Okay, my mic is on. Okay, good. So while Sarah is trying to play the video, I'm going to basically tell you the main difference between a yeast and a mold. So the main difference between a yeast and a mold is that a yeast is a unicellular type of fungi, whereas a mold is a multicellular filaments of fungi. Mold consists of tubular branches with multicellular nuclei. That means it has more than one nucleus. And the reproduction of yeast occurs by budding, whereas mold reproduce by the production of sexual or asexual spores. Does anyone know what asexual reproduction is?
Yes, sir.
Okay, let me hear you.
Is when you need one person to reproduce or one thing to reproduce.
Yes, precisely that what. Yes. So asexual reproduction, it's only one organism involved. Sir, have you found the videos yet?
Yes, I have. You ready? Are you ready now? Are you ready now?
Yes, you can share screen and play it.
Okay.
Today's video is very interesting video and it is fundamental. Here we will discuss difference between yeast and mold. yeast and mold में क्या difference होता है? दोनों ही ये यूकैरियोटिक होते हैं यानी एडवांस्ड हैं विद द सेल न्यूक्लियाई एंड मेम्ब्रेन बॉन्ड ऑर्गेनेली दोनों में होती है बट इसमें डिफरेंस क्या होता है? मेजर डिफरेंस अगर हम देखें डेफिनेशन देखें जीस्ट यूनिसेलर होता है मोल्ड मल्टीसेलर होता है थ्रेड लाइक जीस्ट में बडिंग होती है इसकी फॉर्म देखें जीस्ट के सिंगल सेल में ग्रो करते हैं दूसरा ब्रांच में ग्रो करता है जीस्ट यूनिसेलर होता है मोल्ड मल्टीसेलर होता है हैबिटेट जीस्ट काफ़ी कॉमन है फ्रूट में वेरीज में स्टमक में मैमल के स्किन में भी आ जाता है जो मोल्ड है डैम्प और डार्क ह्यूमिड एरिया में ही मिलता है अपीयरेंस देखें जो जीस्ट होता है वाइट एंड थ्रेडी मोल्ड जो होते हैं फ्यूजी अपीयरेंस होती है एंड कैन बी फाउंड इन सेवरल शेप हाइफे देखें जीस्ट के हाइफे नहीं होते मल्टी सेलर स्ट्रक्चर ही होते हैं इसको सीडो हाइफे बोलते हैं मोल्ड के फिलामेंटस हाइफे होते हैं स्पोर जीस्ट जो है स्पोर नहीं बनाता है मोल्ड के स्पोर होते हैं कॉलोनी मार्फोलॉजी जिसकी जो कॉलोनी है सॉफ्ट होती है ओपेक एंड क्रीम कलर्ड फिलामेंटस होती है मोल्ड की एंड विद द वेजिटेटिव हाइफे कलर जीस्ट जो कलरलेस होते हैं मोल्ड के कलर अलग अलग होते हैं ऑरेंज ग्रीन ब्लैक ब्राउन पिंक पर्पल इंक्यूबेशन रूटीन इंक्यूबेशन टेम्परेचर जो है 25 से 30 होता है इसका 25 30 और 35 होता है और ये टॉलरेट करते हैं वेदर डिटरमिन और बाईफेजिक होते हैं कल्टीवेशन मेथड जीस्ट को ग्रो 24 से 36 आवर में ग्रो किया जाता है और जो मोल्ड होते हैं बहुत स्लोली ग्रो करते हैं जीस्ट जो होते हैं एरोबिक और एनारोबिक कंडीशन में सर्वाइव करते हैं मोल्ड ओनली एरोबिक में जीस्ट जो है 4 से 4.5 pH टॉलरेट कर सकते हैं मोल्ड कैन ग्रो वाइडर रेंज काफ़ी pH वेरिएशन होती है डायग्नोसिस एंड आइडेंटिफिकेशन फिजियोलॉजिक टेस्ट एंड फ्यू की होते हैं इससे इसकी आइडेंटिफिकेशन होती है तो मोल्ड को हम इंटोलॉजी एंड मार्फोलॉजी देखते हैं ई सेक्सुअल स्पोर देखते हैं इनके रिप्रोडक्शन जीस्ट में बडिंग होती है बाइनरी फिशन और इसमें स्पोर बनते हैं ए सेक्सुअल एंड सेक्सुअल ए सेक्सुअल स्पोर जो होते हैं जिसके ब्लास्टोपोर होते हैं और मोल्ड के स्प्रिंजियोस्पोर एंड कोनिडिया होते हैं सेक्सुअल स्पोर जीस्ट में नहीं बनता मोल्ड में बनते हैं जाइगोस्पोर एस्कोस्पोर एंड बेसिडियोस्पोर एनर्जी प्रोडक्शन कार्बोहाइड्रेट को अल्कोहल कार्बन डाइऑक्साइड में कन्वर्ट करते हैं और बाई पॉलीमेरिक सच स्टार्च शेल्वर लिग्निंग सिंपल सब्सटेंस में बनाते हैं मोल्ड। हेल्थ रिस्क, इन्फेक्शन कर सकते हैं जीस्ट इम्यून सिस्टम में। दूसरे मोल्ड एलर्जिक रिएक्शन है, रेस्पिरेटरी प्रॉब्लम करते हैं। अदर रिस्क कंपैरेटिवली लेस, दूसरे ज़्यादा स्पोलेज करते हैं। यूजेस अल्कोहल में, बेकिंग में, विटामिन, सेल स्टडी में। मोल्ड आपको पेंसिलिन देता है चीज़ प्रोडक्शन में। न्यूरोस्पोरा जो है इसको जेनेटिक्स में यूज़ किया जाता है। स्पीसीज़ जिसकी 1500 स्पीसीज़ हैं, तो मोल्ड की 4 लाख स्पीसीज़ है। एग्जांपल देख सकते हो जिसकी सैक्रोमाइसिस, क्रिप्टोकोकस, न्यू फॉर्मिस। और आपके जो मोल्ड हैं एलेंटेरिया, एस्परजिलस, फिसिकैरम, म्यूकर, पेंसिलियम, राइजोफोरस एंड ट्रैकोफाइटोम। So friends, this was all about difference between the yeast and mold. I hope you would have enjoyed this video and very informative. And you can note by the screenshot. Thank you very much.
When you study at University Canada West.
Okay, so that was the end of the video. Does anyone know what people normally do with yeast? That there are some beneficial uses of yeast. Does anyone know of any? What do you think people use yeast to do?
Bake bread.
Yes, that's one of them.
To make wine.
Yes, otherwise known as ethanol. Yeah. So people normally use yeast in to bake bread and to produce rum or wine. Yeah. So similarities between yeast and mold are both yeast and mold are 2 types of fungi. And both yeast and mold contain a sheeting cell wall, means that their cell wall is made up of the similar structure called sheeting. Sheeting is a chemical compound that is found in the cell wall of both yeast and mold. And both yeast and mold are saprophytes, meaning that they are organisms that digest other materials from dead organisms. So we're going to talk about the habitat of a yeast, meaning that where yeast is normally found. So yeast is very common in the environment around you, while molds can be found in damp, dark, or stream-filled areas. And I'm going to talk about the appearance of yeast. You know, the appearance of yeast is oval in shape, and it is colorless and it is smooth. So that is the appearance of yeast. It is oval in shape, and it is colorless and smooth. While mold has a fuzzy appearance, that fuzzy looking thing, it has a fuzzy appearance, and the colors can be green, orange, black, brown, purple, and pink. So that thing that you will see on bread when it is becoming stale or cheese, and most time it look green and fuzzy, that will be the mold. Now, energy production. Yeast converts carbohydrates to alcohol during fermentation, like we had discussed earlier. And mold secretes hydrolytic enzymes to external food sources and absorbs nutrients. Now, hydrolytic enzymes are enzymes that they operate by using water. So those are hydrolytic enzymes that operate in a water environment. Now, to conclude what we've discussed, yeast and mold are two forms of fungi. Both yeast and mold are saprophytes. Can anyone recall what a saprophyte is? Like the same thing I just told you earlier. A saprophyte. Can you recall what a saprophyte is? What is a saprophyte?
An organism that lives in dying or decaying matter.
Yes, yes, yeah. An organism that feeds on the materials of dead organisms. So both yeast and mold are saprophytes, remember that. And they secrete digestive enzymes on the external organic matter to digest them. So saprophytes function, how saprophytes digest dead organism is that they secrete an enzyme on the material that they want to digest. So that's how they feed on other dead organisms, by secreting enzymes. And who remember the names of those enzymes? It starts with H. And they operate in water environments. Who remembers it? The name of those enzymes, it start with H. Any takers? Okay, no, so those enzymes are hydrolytic enzymes, remember?
Pardon, sir?
Do you have red cabbage?
No, I don't have red cabbage.
Okay. Well, hopefully you can get it by Monday, but if not, still log on to the session.
Yes, I'll still try, sir. I'll still try.
Yes, still the session. For this goes for all those who might not be able to get the red cabbage as well. And if you do, remember to prepare the cabbage juice in advance. So when we come, we can have a smooth start, because we don't want to take up too much time. But if you don't get the red cabbage, you can still come and participate in the lab session, and I'll be doing it as well, so you can see the basis of the experiment, because it is just to reveal if some home-based substances, if they are acidic or if they are basic. So the toothpaste that you use, the vinegar, the baking soda, we're going to be testing if they are acidic or if they are basic. So even if you don't get the red cabbage, you can still log on to the session and participate. So I'm going to hand you back over to your coordinator. I'm pretty sure it's February now, and I will see you guys on Monday. Thanks for your time. I hope you learned something.
Yes, bye, sir.
Bye, sir.
All right, guys. I trust that you learned something during your both sessions this morning. You are now on your break. You will return at around 11 for your PowerPoint session. So you can go refresh yourself, go to the bathroom, eat something, go on social media, do what you need to do, and return at 11.
Okay, better.
Should I leave the class?
Yes.
Leave the
You can go.
breakout room.
Completely.
You can leave the breakout room. Mhm.
Okay, guys, you can go for your break and return at 11.
11.
11
Miss?
a.m. Jamaica time, 12 p.m. Eastern Caribbean time. Understand? Understand what I'm saying, Alexandre?
Yes, miss.
It's